Okay, so we'll go ahead and get started. Thank you all so much for joining this evening. I know it's a little bit late in the evening, so I appreciate you being here. My name is Lauren Sheehan, and I'm an application scientist with Lexogen. And today I'm going to be talking to you about one of our most popular products, QuantSeq. And I'm going to introduce you to QuantSeq-Pool, which is one of our newest products in the QuantSeq product family line. And QuantSeq can be used for scalable multiplexing for simplified and inexpensive large-scale gene expression profiling studies. So to start with, I wanted to provide a brief overview of what I'm going to be discussing today. So first, I'm going to give you an introduction to our company, Lexogen, and also to our product portfolio, which is quite extensive. And then next, I'm going to be talking about just comparisons between conventional versus 3' mRNA sequencing, which will lead me into talking about QuantSeq, our solution for 3' sequencing. Following this, I'll be talking about, again, our newest product to the QuantSeq family, QuantSeq-Pool. And then to end the presentation, I'm going to be providing some highlights from a webinar that was given by Dr. Quinn Wills, and this was done last year, which I think provides a very nice showcase to what the QuantSeq technology can be used for. Lexogen is a biotech company that's focused on enabling the analysis of the complete transcriptome. Lexogen was founded in 2007 and is based in Vienna, Austria, specifically at the Vienna Biocenter. We do have a subsidiary in New Hampshire, and this subsidiary was founded in 2014. And as you can tell, our company is made of a multinational team. We also have a worldwide distributor network. So it allows researchers from anywhere across the globe access to our products and our services. And at Lexogen, we have the following mission. We want to empower our customers with innovative, top-quality RNA analysis solutions and support in order to improve the health and well-being for everyone, including our planet. So as mentioned, I want to take you a bit through what we have in our product portfolio. Today I'm going to be focusing specifically on library preparation, but I want everyone to be aware of the end-to-end workflow that we have available at Lexogen. So to start with, I'll talk about sampling. So we have our SLAM-Seq kit, and this is a method for the measurement of newly synthesized and existing RNA in cultured cells. And specifically what SLAM-Seq can do is enables the resolution of RNA synthesis and degradation kinetics. What's not pictured on here is the complete SLAM-Seq portfolio, which includes both Explorer and Kinetic modules. And with these modules, this covers the entire metabolic experiment. So really starting from optimizing the labeling conditions all the way down to labeling nascent and existing RNA for downstream NGS library preparation. So from SLAM-Seq, the total RNA can be used directly into library preparation kits, for example, QuantSeq. And specifically, we have data analysis pipelines that, or we have one data analysis pipeline that's available for analyzing SLAM-Seq QuantSeq data. This is named SLAM-Dunk, and it's currently available on our Bluebee Genomics platform, which I'll touch on a bit later. So now going into RNA preparation and NGS library preparation, so what products we have available. So starting with RNA preparation, we have our split RNA extraction kit, which can be used on a variety of sample types. We have Trapper, which is our small RNA isolation kit. And what Trapper can do is it can isolate risk-associated small RNAs that are found in any organism, tissue, biofluid, and cell type. For ribosomal depletion, we have RiboCop, and we have this for human, mouse, rat, and then also bacterial samples. And then we have polyA selection kit, which is for the rapid and highly specific enrichment of polyadenylated RNAs from total RNA samples. Now going to our NGS preparation kits, we have 4 main kits, and this really depends on the needs and goals of our researcher, which ones they'll pick. So QuantSeq, again, is our gene expression analysis kit, and of course, I'm going to be going into this further in the presentation. For whole transcriptome sequencing, we have our Coral kits. So we have both Coral Total, which is bundled with RiboCop, and then we have Coral mRNA, which is bundled with our polyA selection kit. Luther is another 3' mRNA-Seq kit, but its technology differs quite from QuantSeq, and specifically it's intended for ultra-low inputs as well as single-cell applications. And then finally, for small RNA sequencing, we have small RNA library prep kit, which allows for generation of small RNA libraries from either total RNA or enriched small RNA fractions. We also offer spike-in RNA controls called SERVs. We have our full-length cDNA preparation product called TeloPrime. And then in terms of indexing solutions that we have available, we have both single and dual indexing, and these can be purchased as either add-ons or bundled with our library preparation kits. And Lexogen also offers a sequencing service where customers have the option to send us your RNA, and then we perform library preparation, sequencing, and data analysis. And then finally, I want to touch on several pipelines, NGS pipelines that we have available. And these are hosted on our the Bluebee Genomics platform and on Roslyn. Specifically, we have pipelines available for analyzing data from QuantSeq, Coral, and SlamSeq experiments. And what's really nice about the data analysis tools that we have is any user can utilize them. No prior bioinformatic knowledge is necessary, so they're able to process their own data. And again, today I'm going to be focusing on talking about our QuantSeq technology. So now I'm going to transition over to discussing about the two major types of RNA-Seq. Up until somewhat recently, whole transcriptome sequencing, be it mRNA or total RNA-Seq, is what has been used to look at de novo assembly of transcripts, detecting isoforms, and then also performing expression profiling. So with this technique, you have multiple fragments that are generated per transcript, and you end with a fairly involved bioinformatics analysis, which results in a lot of time and money spent to obtain your NGS results. Now for expression profiling, this is much easier and more straightforward approach because you build your library around the fragment of interest and then simply sequence one fragment per transcript. So because of this, you end with a much simpler bioinformatics analysis, essentially where you're counting the number of sequences and eliminating the need for having to do assembly and normalization. So overall, with this expression profiling method, you can save 1/10 of your sequencing reads because, again, there's no need for mapping to the entire transcript. Because of this, you can multiplex higher and spend less on computational resources due to the simplicity of downstream analysis. So this table is showing more comparison between conventional and 3' mRNA sequencing. So for sample types, high quality RNA is typically what's required for conventional mRNA sequencing, whereas with expression profiling, low quality, including very challenging samples such as FFPE samples, can be processed as well as high quality samples. For the time difference between the two methods, this can also be significant, with 3' mRNA sequencing typically not requiring a fragmentation step. So for the rest of this table, I already covered, so I won't repeat myself, but what I hope you can see are the major advantages of using a 3' mRNA-Seq method, which can save significant amount of time and cost. So this brings me to Lexogen's solution for expression profiling, which is our QuantSeq kit. So QuantSeq started as a single product, which I'm going to cover first, but it eventually branched out into a family of products covering a variety of researcher needs. So in addition to QuantSeq, there's also QuantSeq Flex, which allows targeted sequencing. Today, I'm not going to be going through QuantSeq Flex, so if you have any questions after the talk, you can come up to me and we can chat. QuantSeq-Pool, as mentioned before, is our most recent product release, and I'll, of course, be going into detail about this product in the coming slides. What I want to show now is between QuantSeq and QuantSeq-Pool, both of these kits utilize an oligo-DT primer, which binds to the 3' poly A tail of transcripts and initiates reverse transcription. In QuantSeq Reverse, it's a little different because the researcher has the flexibility of designing their own primers, either for first and or second strand synthesis. So now I want to just share a quick 1-minute video which describes, I think, QuantSeq very nicely.
Welcome to a few minutes of RNA expertise with Lexogen. In this video, we will show you how QuantSeq can simplify your gene expression profiling projects and how you can save valuable time and money. QuantSeq stands for Sequencing That Counts, and is Lexogen's fast, easy, and robust mRNA-Seq library prep that even enables the analysis of challenging material such as FFPE samples. QuantSeq generates only one fragment per transcript at the very 3' end. This enables gene expression profiling with a much lower number of reads per sample and saves sequencing depth for a higher level of multiplexing. This drastically reduces the overall cost per sample. Fragments are generated by priming in the poly-A tail. Only polyadenylated mRNA is channeled into the library prep. No additional pre-processing is necessary, such as mRNA enrichment or ribosomal RNA depletion. Further, QuantSeq successfully generates libraries from degradation-prone input, such as RNA from FFPE samples, since it does not rely on covering the complete length of the transcript. After sequencing, the analysis of QuantSeq data is particularly straightforward and fast. There is no need for isoform identification and length normalization. Reads are simply mapped to the genome and counted for each gene. Are you curious to try QuantSeq?
Okay, so as mentioned in the video, either high or low quality RNA samples, including challenging FFPE samples, can be utilized with QuantSeq. So here I just wanted to go through the QuantSeq workflow with you. So to start, the oligo-DT primer that's used for first-strand synthesis targets and binds to the 3' poly-A tail of transcripts, and reverse transcription proceeds. Importantly, this oligo-DT primer includes a partial Illumina adapter, which is depicted in blue. Following reverse transcription, RNA is removed and random second-strand synthesis is carried out to produce a double-stranded cDNA library. As in first-strand synthesis, a partial Illumina adapter is included in the random second-strand synthesis primer, and in here you'll see this in green. Library generation is concluded with a purification step, and this is there to remove any smaller artifacts or primers that are left over. The library is then amplified by PCR, and this is where complete adapters are incorporated for flow cell clustering, and this is shown by the black and orange bars. Additionally, I7 and/or I5 indices are introduced, and this is depicted by the purple and yellow bars. After library amplification and purification, libraries are then ready for quality control and lane mix preparation. For sequencing, read 1 will generate reads towards the 3' poly-A tail, which will directly reflect the mRNA sequence. And for QuantSeq, we do not recommend paired-end sequencing, as the quality for read 2 will be very low due to reading into the poly-T tail. However, if paired-end sequencing is done, this is okay, because we would just suggest to discard read 2 data and focus on read 1. So up until now, I've discussed the workflow for QuantSeq Forward. However, I want everyone to be aware that we also have another QuantSeq product called QuantSeq Reverse. And with QuantSeq Reverse, it's possible to pinpoint the exact 3' end in read 1. Hence, typically it's used to study alternative polyadenylation sites and for alternative 3' UTR expression studies. With QuantSeq Reverse, paired-end sequencing can be done, but I do want to mention with QuantSeq Reverse, it does require a custom sequencing primer to be used for sequencing, and this custom sequencing primer is provided with our kits. So next I want to touch on the performance of our QuantSeq kit. So as mentioned before, QuantSeq libraries cover the 3' end of transcripts, so you have a significant savings in terms of the read depth that's required. So in the graph to the left, this showcases an experiment where data sets were evaluated for ERCC spike-in abundances. You'll see a major benefit of QuantSeq when plotting the relative coverage across the normalized transcript length. Standard mRNA-Seq distinguishes distributes each reads across the entire length of the transcript. And as you can see, there's underrepresentation of the 3' and 5' ends. And here, just so you can decipher the graph, the gray lines are representing the ERCC spiking controls, and then the areas are representing all other mRNA transcripts. And this is specifically for the mRNA-Seq samples. Whereas with QuantSeq, this is covering only the 3' end of transcripts, and in the graph, this is represented by the blue line, which is our ERCCs, and then the green area, which is the rest of the mRNA transcripts in the QuantSeq sample. The numbers provided are area under the curve, or AUC values, and these are measuring the sequence coverage. So for gene expression and differential expression analysis, 1 read per transcript is sufficient. The additional sequencing space that's gained by focusing only on the 3' end can then be used for a higher degree of multiplexing. So in the present example here, you'll see that the standard mRNA-Seq has around a 12-fold higher relative sequence coverage, which in turn presents the maximal possible reduction in read depth when using QuantSeq, but you're still able to determine gene expression accurately, as you can see on the right. When comparing mapped reads between QuantSeq and conventional mRNA-Seq, focusing on differential expression metrics, you'll see that the results are very similar between the two. You will notice that there's one outstanding feature that's unique to QuantSeq, is that the reads are mapping to no feature to a higher degree compared to mRNA-Seq reads. So this is depicted here by the green bar. And this can largely be attributed to incomplete 3' gene annotation. So reads are indeed mapped correctly to the unannotated 3' UTR and polyA sites, but they're not counted in the protein coding category due to the lack of annotation. So correction of an incomplete annotation or a sensible 3' extension of the gene definition can address the discrepancies between the available annotation and the real sequence transcriptome. So overall, QuantSeq provides you with reliable data to perform gene expression studies. So finally, I want to showcase how QuantSeq can be utilized for low quality, and then here I'll be showing some data on FFPE samples. So this was a very nice experiment that was done using xenograft of a human tumor cell line. And this sample was split into 2 pieces and processed as either a fresh frozen cryo block, so this had a RIN score of 8.3, or embedded FFPE material, which has a RIN score of 2.8. So from these 2 samples, QuantSeq libraries were generated using 50 nanograms of RNA and then sequenced on a HiSeq. So as you can see in the plot to the left, there was a high correlation in gene expression between the 2 libraries with an R-squared value of 0.86. And then regarding gene detection, looking at the middle overlay, and this has a gene detection threshold set at 5 reads per gene, the difference in gene detection was only 3%. So the high concordance of gene expression detected in the cryo and FFPE samples demonstrates how QuantSeq is really the method of choice for gene expression quantification, independent of the RNA input quality that's utilized. So as mentioned earlier, we do have a pipeline for the analysis of QuantSeq data, and this is hosted on the Bluebeam Genomics platform. And I wanted to touch on this because if you were to order a QuantSeq kit from us, you will receive an activation code, which can be used for free data analysis. So for example, if you order a 24 prep kit, you get an activation code that allows you to process 24 FASTQ files through this platform. The Bluebeam platform is extremely user-friendly, and no bioinformatics knowledge is required. The environment is also extremely secure, and it provides users multiple options for uploading and downloading data. And we have a variety of species that are available, but if you find that your reference genome isn't there, you can always contact support, and we can discuss feasibility of uploading this genome for you for cost. Okay, so that concludes my overview of QuantSeq. I hope I've been able to show you that QuantSeq is cost-efficient expression profiling, and it can allow multiplexing of up to 9,216 samples. One thing I didn't touch on is the low input amounts that QuantSeq can handle. So researchers are able to confidently input as low as 1 nanogram of RNA into our QuantSeq kit. We do have protocol modifications available for these lower inputs, and we also have protocol modifications when using low quality and FFPE samples as input. QuantSeq can also allow for targeted RNA sequencing, and this is achieved with our QuantSeq Flex kit. So as briefly mentioned before, a customer can design their own primers for first or second strand synthesis instead of using the oligo-DT primer and the random second strand synthesis primer. And then, as I discussed in the previous slide, QuantSeq data analysis is very simple and straightforward. So you're simply counting gene reads instead of having to do any normalization or assembly. So now I'm going to move on and talk about our newest product, which is QuantSeq-Pool. And as with QuantSeq, I want to start by showing a quick video which provides an overview of QuantSeq-Pool.
Welcome to a few minutes of RNA expertise with Lexogen. Today we will present to you QuantSeq-Pool, Lexogen's most efficient library prep kit for high-throughput expression profiling. QuantSeq-Pool is based on the fast, easy, and robust QuantSeq technology, which we have introduced in another video. With QuantSeq-Pool, you can easily scale up expression profiling projects and save significant time and consumables. In contrast to standard QuantSeq, up to 96 samples are labeled with a unique barcode in the first step. Instead of processing 96 individual reactions, they are now combined by pooling, and all subsequent steps can be handled in one reaction tube. This procedure saves up to 95% of the required consumables in tips, PCR plates, and tubes, and allows handling of large sample numbers. The protocol also reduces the technical variation between samples because they are all treated the same way at the same time. Each pool of up to 96 samples can be additionally labeled with unique dual indices, which allows multiplexing of several pools. With up to 384 UDIs, this enables simultaneous analysis of up to 36,864 samples. As with QuantSeq, QuantSeq-Pool also allows fast and easy data analysis by straightforward read counting. Are you curious to try QuantSeq-Pool?
Okay, so as with QuantSeq, I'm going to go through the QuantSeq-Pool workflow. And as you can tell from the video, QuantSeq and QuantSeq-Pool have very similar methodologies, and what I'll do now is just point out some differences between the two. So first, in the oligo-DT primer used for first-strand synthesis, there's a sample barcode that's present. So this is indicated here by the blue arrow, and this is our I1 barcode. Additionally, there's also a unique molecular identifier, which is here in red. So following reverse transcription, samples are then pooled into one reaction, allowing for streamlined processing. So this eliminates the need for having to QC individual libraries and saves researchers a significant amount of time. So as with QuantSeq, QuantSeq-Pool generates reads towards the poly A tail and also utilizes standard sequencing primers. However, with QuantSeq-Pool, this does require paired-end sequencing, at least asymmetrical paired-end sequencing, in order to obtain the I1 and UMI sequence information. I7 and I5 indexing is optional, and this is due to the presence of the I1 sample barcode included in the first-strand synthesis. So ultimately, the presence of these additional indices are dependent on how many samples you plan to multiplex. So currently, we have 96 unique I1 barcodes. So if a user is planning to multiplex more than 96 samples, then additional indexing would be required. The most important takeaway with QuantSeq-Pool is this allows you to pool up to 96 samples into one tube. Because this pooling is done very early in the protocol, it allows for a very simplified workflow, helping to keep things consistent and efficient. Moreover, the scalable throughput and multiplexing is much higher than with our standard QuantSeq kit. So here are just a few examples of what can be done in terms of what can be multiplexed. And as you can see all the way to the right, with the use of our 384 unique dual indices, up to 36,864 samples can be processed with QuantSeq-Pool. So now I'll move on to talking about the performance of our QuantSeq-Pool kit. So QuantSeq-Pool enables robust and consistent gene expression even at low sequencing depth, which is shown here. So in this experiment, there were 8 replicates of QuantSeq-Pool libraries, which were prepped with 10 nanograms of UHR and sequenced on a NextSeq. Gene detection was analyzed at several read depths. So on this graph here, 100,000 reads per sample is shown in gray, 500,000 reads per sample is in green, and then 1 million reads per sample is in blue. The number of detected genes was counted at a threshold of over 10 counts per million, or CPM, which allows you to see highly expressed genes. And you'll see that at only 1 million reads per sample, 9,000 genes can be detected. And if you were to exclude everything with a CPM below 1, you can get up to 14,000 genes detected, but it really all depends on the read depth that you utilize with how many genes you can detect. As you can see, between the replicates, the number of genes detected is extremely consistent. So when comparing QuantSeq and QuantSeq-Pool, there is a high correlation between the two methods when looking at gene expression. So here, what we're looking at is a correlation analysis on the left and then an overlay on the right. So in the left correlation analysis, you'll see QuantSeq on the y-axis and then QuantSeq-Pool on the x-axis, and they have very similar gene expression profiles with an R-squared value of 0.9. In the right Venn diagram, QuantSeq-Pool is depicted in blue. QuantSeq is depicted in blue, and then QuantSeq-Pool is depicted in light green. So there's very nice correlation, but one thing I want to note is the differences that you see are due to mitochondrial transcripts, and this is likely attributed to the modifications that are used in our QuantSeq protocol for low input samples. But overall, you can see that QuantSeq-Pool and QuantSeq perform very nicely. And then here, this is just showing high correlation, again, between replicates. And you can see between QuantSeq-Pool on the left and QuantSeq on the right that there's very high correlation between them. So next, I want to show a very nice experiment that was done by our R&D team to show the performance of QuantSeq-Pool in terms of differential expression analyses. So to mimic differential expression, a spike in RNA control called SERVs were added to our UHR samples at different known amounts. So in this way, the results would be predefined upon cross-comparison, and we would know what we call here the theoretical log2 fold change, or theoretical LFC, and this can be seen on the x-axis. The observed LFC is the result of the experiment, and this is depicted on the y-axis. So if the plot results in a diagonal trend, then this would indicate that input matches output. So this box plot is showing QuantSeq, which is in green, and then QuantSeq-Pool, which is in blue. And you'll see that the expectation and the results correlate very nicely for both kits. QuantSeq-Pool is slightly closer to what's expected, and also the wideness of the bars is a bit reduced, and this can be contributed to the batch processing. So when you have all 8 replicates in one tube, versus with QuantSeq, you're processing these samples independently. So overall, this shows that QuantSeq-Pool can indeed be used for accurate differential expression analyses. So now I'll move on to covering the benefits of QuantSeq-Pool. And one thing that I've already mentioned is the amount of time that can be saved by performing this early pooling. So there will be time saved during library generation and amplification, but more importantly, a significant amount of time is going to be saved during QCing and preparing lane mixes. So if you're processing 96 samples using conventional mRNA-Seq, approximately 11 hours could be dedicated to QCing and normalizing individual libraries. With QuantSeq-Pool, again, if you have 96 samples, you're only dealing with one tube. It would technically be possible to go from preparing your samples to sequencing in just one day. The other major benefit with QuantSeq-Pool is cost savings. So this is showing the benefits of pooling, and again, instead of having to process 96 samples independently, you're just processing one sample. So doing this, you can save a lot on plasticware, such as PCR plates, pipette tips, and so on. Additionally, this is going to eliminate the need for individual QCing. So if preparing 96 samples for QCing for an mRNA-Seq experiment, running all these samples on a fragment analyzer can add up very quickly. With QuantSeq-Pool, again, you would only need to QC one sample, and underneath this one tube, there's 96 individual libraries. In the graph to the right, here are just the cost of QuantSeq-Pool versus conventional mRNA-Seq. And importantly, no sequencing or kit costs are included here. So it's only the hidden costs, which can be considered as plasticware and QC reagents. So compared to conventional mRNA-Seq, QuantSeq-Pool can save a significant amount of time and consumable costs. But how do you know which QuantSeq kit would be right for you? So we have this really nice figure on our QuantSeq-Pool webpage, which will help you decide if you should go with standard QuantSeq or if you can use QuantSeq-Pool. Currently, there are some limitations with QuantSeq-Pool, and specifically this is related to the RNA quality, the sample type, and the input amount that you have. So if the RNA quality is medium to high, so typically I'll tell my customers a RIN score of 6 or more, this can be processed with QuantSeq-Pool. If a customer is dealing with FFP samples or just very low quality samples, I would instead direct them to QuantSeq. For sample types, the samples do have to be homogeneous. If the customer is dealing with a very diverse set of samples that ideally requires individual QCing, this is where QuantSeq would be fit for them. And then finally, QuantSeq-Pool has an input amount of between 10 to 120 nanograms per sample. So if a customer is dealing with extremely low inputs or they have a need for going above 120 nanograms per sample, they would instead go with standard QuantSeq. So as long as you have medium to high RNA quality, your samples are homogeneous, and then you have an input between 10 to 120 nanograms per sample, QuantSeq-Pool can be utilized. So this concludes my overview of QuantSeq-Pool, and I encourage you all to visit our website and then also come talk to us if you have any questions about the product. And here I'm just going to summarize some of the benefits that I covered. So QuantSeq-Pool encompasses all of the benefits of QuantSeq, but it allows users to save time and then also allows them to scale up the amount of samples that they would like to multiplex. So going from 96 samples to 36,864 samples. So due to the early pooling, you're able to save a significant amount of cost associated with lab consumables and sequencing costs as well. Additionally, early pooling allows you to process potentially hundreds of samples in a single day. And then finally, as with QuantSeq, QuantSeq-Pool data analysis is very straightforward and requires simple counting. So I want to mention, right now we do not have a QuantSeq-Pool pipeline available on our Bluebee platform, but we do have this available on GitHub. Okay, so to conclude my presentation, I wanted to summarize some key points from a webinar that was given by Dr. Quinn Wills at Okra Bio. And what he did is he utilized QuantSeq for sequencing 1,000 donor livers. The focus of Dr. Wills' research is utilizing deep phenotyping to develop RNA therapies for liver targets of chronic disease. And the full webinar can be found on our webpage. So Dr. Wills' group studies liver fat disease called NAFLD, and NAFLD stands for non-alcoholic fatty liver disease. So the top part of the slide is showing a very simplified model of disease progression with systemic and liver-specific consequences. Okra Bio was founded to develop therapies for all of the challenges associated with liver disease, specifically addressing the two biggest challenges which are shown below. The first challenge is with NAFLD, it's a silent disease where most patients with psoriasis won't even know that they have it until it's too far along. This means that there's very little data on disease progression and it's very difficult to study. The second challenge is NAFLD is complex. So it not only has cardiometabolic interactions, but it can also interact with more cell types and pathways later on. So as progression proceeds, it's less likely that a single therapeutic target will be effective in treating the disease. So to tackle these challenges, Dr. Wills is using deep phenotyping, which can be used to understand the biological details which are crucial to understanding a chronic disease like NAFLD. So as shown here, deep phenotyping can be thought of as a marriage between genetics, imaging, cellular genomics, and statistical and machine learning data to model the information they have. It is the generation of tissue information mapped to genes and clinical phenotypes that will help fill in gaps. In the webinar, Dr. Wills then goes to discuss a collaborative project with transplant surgeons at the University of Oxford, where they studied 1,000 donor livers to obtain a better understanding of the silent years of NAFLD. So here are two pictures, or here's one picture of two liver biopsies. And the blue areas here are denoting some cells that are undergoing something called ballooning degeneration. What Dr. Wills wanted to do was overlay imaging with spatial RNA sequencing, and this is where our QuantSeq kit comes to play. As these were particularly challenging biobanked FFPE samples, Dr. Wills collaborated with our Lexogen Services team to develop a custom workflow for lysing the cells to extract the RNA, and then he was able to use the RNA to prep libraries with QuantSeq. So with this experiment, his group was able to ask three very specific questions. So first, how disease niches compared to less disease niches, how niches compared across donors, and then how gene expression changes map to 150 clinical variables. So here's just one example of the output from their secondary model, which demonstrates the validity of the data that they generated. An engine software was utilized which contains over 250,000 sets, which will help them contextualize the data. So what Quinn did is he asked the software which data sets most closely resembled the signature of genes associated with percentage fat in the livers from this current experiment. The software presents the above network where each point is one data set. The top 10 hits have been denoted with black points in their centers, and they've also been annotated. So these points are phenotypes from the study which are tightly correlated with percentage fat. You'll also see that some of the top hits are gene signatures extracted from published studies of liver fat accumulation. So overall, what they were able to see with their study is similar to what others have seen, but even more. So not only because of the statistical power that was used, but because Quinn's group used spatial information to find genes which others may have previously overlooked. So here's an example of another top-ranked gene. On the left, what you'll see are three examples of histological association. So what you can see is this gene is positively associated with key disease histology phenotypes. So we're looking at percentage lipid, vesicle size, and then also ballooning score. Clinically, it demonstrates a positive association with both girth and blood gamma GT. However, they found that this gene had an inverse relationship with C-reactive protein, which is a measure of inflammation. And this inverse relationship was quite surprising to them. They weren't expecting it. And when they broke down the data spatially, specifically looking at the gene's expression in healthy tissue, which is in green, fatty tissue, which is in yellow, and then tissues that are beginning degeneration and inflammation, which is in red, the latter niches observed the same reverse association with inflammation, which is shown on the x-axis in immune cell count. So this gene is particularly interesting to them. They're currently studying this gene, and they think that it could play a very important therapeutic role in managing liver-specific complications of NAFLD. So in the webinar, Quinn goes on to discuss how this type of data generation is really only the beginning to building actionable models. So they refer to this process as their 5 Cs. Charting, which is what they did here, is the curation and generation of rich data, which allows for target discovery. The next 3 C's happen in their Oxford labs and maps to what other target validations might also do. First, it's to clearly establish a gene's causality with one or more stages of the disease. Next, it's to understand the complexity, which means to determine what other genes it's interacting with. And then finally, you want to know its coherence, which is, in the simplest case, making sure that it is acting similar in different models. So they confirm therapeutic efficacy in perfused human livers and sequence, so they can eventually compare all data to the greater body of data that's available. So with that, I'll wrap up my very brief summary of Dr. Will's work. Again, I encourage you to check out the webinar. He goes into much more detail on this project, and then also what his lab is doing to study liver disease. So I want to thank you all again for attending this technology showcase, and if you have any questions, I would be more than happy to answer them. Thank you. Yeah.
Just out of curiosity, how low in terms of, I guess, quantity can you go with FFP and QuantSeq?
Yeah, so you can go pretty low. I believe that we have validated the kit with 500 picograms, if not less. Yeah, so you can go very low and still get good data from it. Mm-hmm.
I think one of your earlier slides, you had a Venn diagram showing like gene detection with, I think it was a CPM, was it counts per million on that slide? And then there was a relatively large difference between the FFPE and fresh frozen.
For the 1 count, yeah.
For the 1 count, what do you think those are?
So it may just be differences in the intact samples versus the degraded samples, what's actually picked up by the kit. I would have to go back and look through the data to see exactly what that was, but I think it's just the difference in the degradation of the RNA and what's actually picked up with the kit.
Do you think any of them, any of those, like, I don't know, 5,000 or so could be legitimate genes? Like if you looked in that list, do you think you would see any genes that you like recognize?
Are you talking about for the freshly frozen samples compared to? I mean, yeah, I would assume so, but I haven't gone through the data in too much detail to know what's actually picked up. Yeah. But again, so that would be really just detecting everything under the sun with just having 1 count per gene. Yeah. So you may miss those with FFPE samples, the very lowly expressed ones. So I would expect that it would be more highly expressed ones that are represented there. Mm-hmm. Okay. Well, thank you again for joining, and if there's any other questions, we'll be here for a little while so we can chat.